The current chapter involves fatty acid catabolism. These are the learning goals for this chapter. This chapter will give you a fairly good description as to how fats are digested in animals. To understand this, you need to understand as to how fats are mobilized and transported in tissues. In addition, you also need to understand how fats are oxidized. By the end of this chapter, you would have learned about chemical moieties called as ketone bodies as to what these are and how they are produced. There are three sections to this chapter. The first one is digestion, mobilization, and transport of fats. Here, we will discuss the source of fatty acids and the routes by which they travel to the site of their oxidation. We will then look at the chemical steps of fatty acid oxidation in mitochondria. The complete oxidation of fatty acids to carbon dioxide and water takes place in three stages. The oxidation of long-chain fatty acids to two carbon fragments in the form of acetyl-CoA is called as beta oxidation. As you all know from your basic biochemistry course that the oxidation of acetyl-CoA to carbon dioxide happens in the citric acid cycle, and as a result, the electrons that are generated gets transported or transferred into mitochondria to initiate the electron transport chain. We will briefly look at how electron transport happens when fats are oxidized, specifically in terms of beta oxidation. And we will conclude the chapter with a description of an alternative fate for the acetyl-CoA molecule formed by beta oxidation and the production of ketone bodies in liver. So, the first section is digestion, mobilization, and transport of fats. Oxidation of fatty acids is a major energy source in many organisms. About 1/3 of our energy needs comes from dietary triacylglycerols. As you may know, triacylglycerols are formed from fatty acids. Glycerol is when substituted with three fatty acids forms triacylglycerols. Triacylglycerols provide more than half the energy requirements of some organs, particularly the liver, heart, and resting skeletal muscle. Many hibernating animals, such as grizzly bears, rely almost exclusively on fats as their source of energy. In vertebrates, specialized cells called adipocytes or fat cells store large amounts of triacylglycerols or fat droplets that nearly fill the cell. Triacylglycerols are neutral fats. If you look at the figure here, it shows a cross-section of human white adipose tissue. Each cell contains a fat droplet that is shown in white, and it is so large that it squeezes the nucleus against the cell membrane. So, every time you see or hear adipose tissue, it stores fat. Fats provide efficient fuel storage. Fats have advantages over polysaccharides as fuel storage, and that's because of these two reasons. Fatty acids carry more energy per carbon because they are more reduced. Fatty acids are often less solvated and carry less water because they are nonpolar. This is the structure of a triacylglycerol. Triacylglycerol or triglycerides are neutral fats, and they contain fatty acids that are highly reduced, which make them especially suitable as a storage fuel. The long alkyl chains of the fatty acids in triacylglycerol are essentially hydrocarbons. They're highly reduced structures with an energy of complete oxidation more than twice that for the same weight of a carbohydrate or a protein. This gives triglycerides a very high advantage to be used as fuel storages. The advantages of triacylglycerols are compounded by their extreme insolubility in water. One of the main reasons why triacylglycerols are insoluble is because of the long hydrocarbon chain of these fatty acids. In addition, triacylglycerols aggregate in lipid droplets, which do not raise the osmolarity of the cytosol, and they are unsolvated, which means that triacylglycerols that are formed from individual fatty acids do not hydrogen bond with water molecules. In contrast, water of solvation can account for 2/3 of the overall weight of stored polysaccharide molecules. Now, if you were to look at the structure of a polysaccharide like amylose or a disaccharide like sucrose, you can see that these molecules have hydroxy groups attached to them. These hydroxy groups could in turn hydrogen bond with water molecules, forming a hydration shell. And that could account for 2/3 of their overall weight. And that makes them not so good for storage purposes. So, as a result, monosaccharide like glucose or polysaccharide like glycogen are used for short-term energy needs and quick delivery. Fats, on the other hand, are stored for long-term energy needs. They're really good for storage and for slow delivery. This slide gives a pretty good description as to how fats are mobilized in the body. Dietary fats are absorbed in the small intestine, but before they are absorbed through the intestinal wall, they must be converted from insoluble macroscopic fat particles to finely dispersed microscopic micelles. This solubilization is carried out by bile salts that are synthesized in the liver and stored in the gallbladder. These bile salts are released into the small intestine after the ingestion of a fatty meal, as you could see. Bile salts are amphifatty compounds that act as biological detergents, converting dietary fats into mixed micelles of bile salts and triacylglycerols. Micelle formation enormously increases the fraction of lipid molecules accessible to the action of water-soluble lipases, in the sense that you need to convert these triacylglycerols into smaller particles like diacylglycerols or monoacylglycerol or the fatty acids and glycerols. And for this purpose, lipases need to act on triacylglycerols. Once lipases convert these triacylglycerols into smaller particles, these are then taken up by the epithelial lining on the intestinal surfaces, or what is called as the intestinal mucosa. When they're reconverted in the intestinal mucosa, these smaller aggregates are reconverted into triacylglycerols and packaged with dietary cholesterol and specific proteins into lipoprotein aggregates called as chylomicron. Apolipoproteins, as they call them here, are lipid binding proteins, and they're specifically found in blood and are responsible for the transport of triacylglycerols. Now, apolipoproteins, what apolipoprotein means is that a protein that is free of lipid. Once an apolipoprotein binds a lipid, it's called as a lipoprotein. So as you could see, once the chylomicron, which composes of these triacylglycerols and the lipoproteins, they move into the bloodstream. The protein moieties of lipoproteins are recognized by receptors on certain cell surfaces. In lipid uptake from the intestine, chylomicrons, which contain apolipoprotein C2, move from the intestinal walls and into the bloodstream, which carries them to muscle and adipose tissues. So as you know, adipose tissue is where fats get deposited. So these chylomicrons get carried through the bloodstream into the adipose tissues. And once they are carried into the adipose tissues, they are also carried to muscle tissues. In the muscle, fatty acids are oxidized for energy. In adipose tissues, they are re-esterified for storage. That's the difference between two different cells as to how fats are being treated. Right? Now, once these are deposited in specific cells or carried to specific organs, the remnants of chylomicron, whatever is left behind, that are depleted of most of their triacylglycerols, but still containing cholesterol and apolipoproteins. This remnant travel in the blood to the liver. Right? It's carried to the liver where they are taken up by endocytosis. Triacylglycerol that enter the liver by this route may be oxidized to provide energy or to provide precursors for the synthesis of ketone bodies. Shown here is the molecular structure of a chylomicron. As you may know from the previous slide, chylomicrons are being used to transport lipids through the bloodstream into various tissues. Now, the components of chylomicron are essentially apolipoproteins, as shown here. The names are B48, C2, or C3. The different apolipoproteins, and about 80% of chylomicrons are composed of triacylglycerols and cholesterol esters. Now, the makeup of chylomicron has phospholipids on the surface layer that is exposed, facing the aqueous phase. The triacylglycerols are sequestered in the interior, making up, like I said, more than 80% of the mass. And several apolipoproteins, they protrude from the surface, and they act as signals in the uptake and metabolism of chylomicron contents. This slide gives a pretty good description as to how hormones trigger mobilization of stored triacylglycerols. We have seen as to how neutral lipids are stored in adipocytes in the form of lipid droplets. When low levels of glucose in the blood trigger release of hormones, this glucagon. Glucagon binds to a receptor, which is a G protein coupled receptor. And we all know as to how G protein coupled receptor works. Once GPCR is activated, the GS protein further activates adenylyl cyclase, an enzyme that triggers the formation of cyclic adenosine monophosphate, or CAMP. CAMP then further activates pKa, or protein kinase A. In this case, protein kinase A phosphorylates two proteins. One of them is a hormone-sensitive lipase, or HSL. The second protein is called as a perilipin. A perilipin is found in adipocytes as bound to another protein called a CGI58. So perilipin bound to CGI58 is what is responsible for restricting access to lipid droplets, preventing lipid mobilization in the cells. Once pKa phosphorylates perilipin dissociates from CGI58. Now, once CGI58 is dissociated from perilipin, it activates another protein called ATGL. And once perilipin is phosphorylated, it interacts with hormone-sensitive lipase that is phosphorylated. So phosphorylated perilipin interacts with phosphorylated HSL. Now, the first step, ATGL, what it does is it converts triacylglycerols, that are fats that are stored in this lipid droplet. ATGL converts triacylglycerol into diacylglycerol, which means one fatty acid is being released or hydrolyzed. The HSL that is being activated further converts diacylglycerol into monoacylglycerol. So another fatty acid is released. Another protein called as monoglycerol lipase, or MGL, further converts monoacylglycerol into fatty acid and glycerol moiety. Now the result is you get 3 fatty acids from one triacylglycerol moiety. These fatty acid molecules then exit the adipocyte into the bloodstream, where it is taken up by protein called as serum albumin. Serum albumin transports fatty acids all the way into a myocyte. A myocyte is a muscle tissue or a muscle cell in this case. And these fatty acids enter a myocyte through fatty acid transporter protein. Now, why is this important? Because you need energy. And in the energy crisis, during energy crisis, the fatty acid is being converted to energy by forming ATP, and this happens by a beta-oxidation pathway where a fatty acid is converted to carbon dioxide. Now we'll talk about this transport pathway later on. In this slide, I would like to draw your attention to some of the key players involved in fat mobilization and transport. One of the key players is called as apolipoprotein. Like I said before, these are proteins in lipid-free form and are lipid-binding proteins in the blood that are responsible for the transport of triacylglycerols, phospholipids, cholesterol, and cholesterol esters between organs. The second key player are called lipoproteins, which are formed when apolipoproteins combine with lipids to form several classes of lipoproteins. We have seen this before, but I just wanted to grab your attention to some of these key points. The third one is a lipoprotein lipase. So like I said, a lipase is something that hydrolyzes a lipid, right? Now in this case, a lipoprotein lipase hydrolyzes triacylglycerols to fatty acids and glycerol. Now, there are various different kinds of lipases that are involved in fat transport and mobilization. And like we saw in the previous slide, one of the key players is the Adipose triacylglycerol lipase, the hormone-sensitive lipase, and the monoacylglycerol lipase. Why these three? Because these three have different functions. And ATGL converts a triglyceride to a diglyceride, or a triacylglycerol to a diacylglycerol. Whereas a hormone-sensitive lipase converts a diacylglycerol to monoacylglycerol. And a monoacylglycerol lipase converts monoacylglycerol to a free fatty acid and glycerol. So now you have to remember the pathway that we saw in the last slide. The lipids that are stored in adipocytes as lipid droplets, once they exit the adipocyte, they are exiting as free fatty acids and glycerol. About 95% of the biologically available energy of triacylglycerols reside in their 3 long-chain fatty acids. Only about 5% is contributed by the glycerol moiety that is being produced. The glycerol released by lipase action is phosphorylated by an enzyme called as glycerol kinase. And so what it does is glycerol kinase converts glycerol to glycerol 3-phosphate using ATP. Now, once glycerol 3-phosphate is formed, another enzyme called glycerol 3-phosphate dehydrogenase converts glycerol 3-phosphate to dihydroxyacetone phosphate. Now, once dihydroxyacetone phosphate is formed, another enzyme called a triosphosphate isomerase converts dihydroxyacetone phosphate to D-glyceraldehyde 3-phosphate. We have seen these reactions in the glycolysis pathway. So, glycerol that is produced in excess enters glycolysis using these 3 reactions. Once these fatty acids enter a myocyte, they are activated and transported into the mitochondria. The enzymes of fatty acid oxidation in animal cells are located in the mitochondrial matrix, and hence these fatty acids need to enter mitochondria to undergo beta oxidation. The fatty acids with chain length of 12 or less can enter mitochondria without the help of a membrane transporters. Those with 14 or more carbons, which constitute the majority of free fatty acids that are obtained in the diet or released from the adipose tissues, cannot pass directly through the mitochondrial membranes. These fatty acids that are 14 carbon or more must undergo the 3 enzymatic reactions of what is called as a carnitine shuttle. So, this involves the first step of activation of fatty acids. So fatty acids first get converted to fatty acyl-CoA, and this is catalyzed by fatty acyl-CoA synthetase, and that's the enzyme here. So this enzyme has 2 substrates. One is a fatty acid, right? Now this fatty acid more likely has 14 or more carbon atoms, and it uses ATP. So first step that it catalyzes is it converts a fatty acid into a fatty acid adenylate. We all know what a fatty acid adenylate is. It uses ATP, and the carboxylate attacks this phosphate, and the pyrophosphate moiety is released from this. Now once this reaction happens, you get a fatty acid adenylate, which is enzyme bound, remember. Now in the next step, the thiol of coenzyme A attacks the acyl-adenylate. So CoASH, this thiol is a free thiol, it's a nucleophilic thiol, and it attacks this specific carbonyl carbon, and it pushes out the adenylate because it's a good leaving group, right? Displacing the AMP and forming the thioester. And this thioester is called as fatty acyl-CoA. And if you were to look at the energetics in this step, the whole step has a negative free energy of minus 15 kilojoules per mole for 2 steps. And the next energetically favorable process is conversion of pyrophosphate to 2-phosphate molecules. And this is catalyzed by the enzyme inorganic pyrophosphatase. And the delta G 0 prime for this reaction is about minus 19 kilojoules per mole. So the total energetic contribution is the sum of this and this. So that's 15 minus 19, that is minus 34 kilojoules per moles. This slide basically reiterates as to what I have said before. Fats are degraded into fatty acids and glycerol in the cytoplasm of adipocytes. We have seen this before, right? Fatty acids are transported to other tissues for fuel throughout the blood. And for fuel, you need to convert fatty acids to carbon dioxide, and electrons need to be released. We'll talk more about this a little later. So that process is called beta oxidation. Beta oxidation of fatty acids occurs in the mitochondria, like I said before. So small, less than 12 carbon fatty acids diffuse freely across mitochondrial membranes. However, fatty acids that have more than 12 carbons are larger fatty acids. Most free fatty acids are transported via acyl-Carnitine or carnitine transporter. And as shown here is the structure of carnitine because the fatty acids usually get attached to this molecule, the hydroxyl group of carnitine. And this is what carries these fatty acids into the mitochondrial matrix. Fatty acyl-CoA esters formed on the cytosolic side of the outer mitochondrial membrane can be transported into the mitochondrion and oxidized to produce ATP, or they can be used in the cytosol to synthesize membrane lipids. Now, this is the fatty acyl-CoA ester that was synthesized from a fatty acid, right? Fatty acids destined for mitochondrial oxidation are transiently attached to the hydroxyl group of carnitine. Now, this is carnitine, and this is the hydroxyl group of carnitine, and the enzyme called as carnitine acyl transferase attaches acetyl-CoA to this hydroxy group of carnitine to make this acyl-carnitine molecule. The transesterification happens once the acetyl-CoA molecule, the fatty acyl-CoA molecule, moves across the outer membrane of the mitochondria. This is where carnitine acyl transferase 1 is located. And carnitine is found in the intermembrane space. And so this protein attaches fatty acyl-CoA to carnitine once it moves across the membrane. In the inner membrane space, the fatty acyl-carnitine ester diffuses through the intermembrane space and enters the matrix. And this happens via the action of a passive transport through this transporter called as acyl-carnitine or carnitine cotransporter. This cotransporter moves one molecule of carnitine from the matrix to the intermembrane space, while it moves one molecule of acyl-carnitine from intermembrane space into the matrix. In the third and final step of the carnitine shuttle, the fatty acyl group is transferred from carnitine to an intramitochondrial coenzyme A. So the CoA is also present in the matrix, and this fatty acyl-CoA fatty acyl group is transferred to another CoA and forms fatty acyl-CoA in the matrix. So the three steps are converting the fatty acyl-CoA into carnitine, acyl-carnitine, so that it can be transported across the intermembrane space into the matrix. And once it is in the matrix, acyl-CoA, the acyl-carnitine molecule is converted back into acyl-CoA. The second section involves fatty acid oxidation. Fatty acid oxidation occurs in the mitochondria in three stages. Stage 1 consists of oxidative conversion of 2 carbon units into acetyl-CoA via a process called as beta-oxidation. This happens with concomitant generation of NADH and FADH2. This involves the oxidation of beta carbon of the fatty acid to thioester of fatty acyl-CoA. Stage 2 involves oxidation of acetyl-CoA into carbon dioxide via the citric acid cycle with concomitant generation of NADH and FADH2. Stage 3 generates ATP from NADH and FADH2 via the respiratory chain. Let's look at these three stages of fatty acid oxidation in a little detail. In the first stage, fatty acids undergo oxidative removal of successive 2 carbon units in the form of acetyl-CoA. Now, in the case of a 16 carbon palmityl CoA, this undergoes 7 passes through the oxidative sequence. As you can see, this is 1, 2, 3, 4, 5, 6, 7. At the end of the 7 cycles, right? You're left with two carbons, carbon 15 and 16. And after each pass, it loses two carbon molecules in the form of acetyl-CoA. So there's one acetyl-CoA coming off 2, 3, 4, 5, 6, 7, 8. So in total, you get 8 2-carbon acetyl-CoA molecules. Right. Now, the formation of each acetyl-CoA requires removal of 4 hydrogen atoms from the fatty acyl moiety by enzymes called as dehydrogenases. Now, these dehydrogenases, what they do is when they convert this molecule into individual acetyl-CoA molecule, they make NADH or FADH2. In the second stage of fatty acid oxidation, the acetyl groups of acetyl-CoA are oxidized to carbon dioxide in the citric acid cycle. So you have seen citric acid cycle in the biochemistry course. And you know that you get 2 carbon dioxide per cycle. So for 8 acetyl-CoA that enter citric acid cycle, you get 16 carbon dioxide and 64 electrons from this citric acid cycle. The first 2 stages of fatty acid oxidation produce the reduced electron carriers NADH and FADH2, comes from stage 1 as well as stage 2, and it enters third stage by donating electrons to the mitochondrial respiratory chain. It's also called as the electron transfer chain, right? And this electrons is passed to oxygen with the concomitant phosphorylation of ADP to ATP. The energy released by fatty acid oxidation is thus conserved as ATP. Let us take a closer look at the first stage of fatty acid oxidation, beginning with the simple case of a saturated fatty acyl chain with an even number of carbons. A classic example is palmitoyl-CoA that we looked at before. It has 16 carbon atoms and it is a saturated fatty acyl-CoA. Once palmitoyl-CoA enters the mitochondrial matrix, an enzyme called as Acyl-CoA dehydrogenase acts on it and oxidizes palmitoyl-CoA to trans delta 2 enoyl-CoA. Now this is how it does it. It uses FAD as a cofactor and reduces FAD to FADH2. And at the same time, it oxidizes palmitoyl-CoA by introducing a double bond between the alpha and the beta carbon. Now the new molecule that is formed is called as a trans delta 2 enoyl-CoA, and as the name suggests, it has a trans double bond, unlike the naturally occurring cis fatty acids. Now, once this molecule is formed, it is acted upon by an enzyme called as enoyl-CoA hydratase. As the name suggests, it uses water molecule and adds the water molecule to the beta carbon across the double bond to form L-beta hydroxyacyl-CoA. So the first step is a dehydrogenation reaction, and the second step is a hydration reaction. Once L-beta hydroxyacyl-CoA is formed, it's acted upon by an enzyme called as beta hydroxyacyl-CoA dehydrogenase. What this does is it oxidizes L-beta hydroxyacyl-CoA to beta ketoacyl-CoA, and at the same time reduces NAD plus to NADH. So after 3 reactions, you get 2 plus 2, 4 electrons from the 2 dehydrogenation reactions. And these 4 electrons combined enter the respiratory chain. Now, it doesn't end here at beta ketoacyl-CoA. Once this molecule is formed, it is being acted upon by Acyl-CoA acetyltransferase or thiolase, an enzyme that uses coenzyme A as a cofactor and splits beta ketoacyl-CoA into 2 molecules. One is a Myers-Doyle-CoA and the next is acetyl-CoA. So this is the first acetyl-CoA that is coming out of the 7 beta oxidative steps. So this is the first beta oxidative steps that involves 4 enzymes. So there are 7 steps, and all these steps involves these 4 enzymes. And what happens is myristoyl-CoA, which has now 14 carbon, will get converted to smaller carbon units.